Hello, everyone, and welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It is my great pleasure to introduce our speaker today, Dr. David Matuskey. Dr. David Matuskey is a board-certificate psychiatrist and neuropsychiatrist with research work that has been translational in nature and focused on elucidating the underlying pathology of brain conditions such as autism spectrum disorder, substance abuse, and neuropsychiatric disorders such as Parkinson's and Huntington disease, with an aim to find effective clinical treatment guided by molecular neuroimaging. Studies have included using in vivo PET imaging to investigate the role of receptors such as dopamine D3, serotonin 1B and 6, and mGluR5, and kappa opioid, neuroinflammation, and most recently, synaptic density in clinical and non-clinical populations. Ongoing interests include imaging neuropsychiatry and addictive disorders and the demographic, social, and environmental factors influencing our brain. I would like to remind our audience to please address any questions using Q&A box or raise their hand. And Dr. David Matuskey, thank you very much for coming today. The virtual stage is yours.
Thank you so much for the introduction. It's very kind of you and really honored to be with you guys over at Martinos. We're friends across the state lines here. Even I just realized I haven't been there yet. So at some point I'd love to come over there and see what you guys are up to. Okay, so I was thinking of a good connection, a Yale-Harvard connection. And there's one really here with Harvey Cushing. And so Harvey Cushing was over at Harvard years ago, of course, and then retired. I think he was actually an undergrad and then ended up retiring over here at Yale. And so he was the father of neuroscience, and in some ways just of some clinical neuroscience in general. And so this is actually an older picture I got online from the basement of Harkness dorm. So this is the medical student dorms. And from what I was told, there was like a rite of passage where, somewhere in their early years, they would have to run down here in the basement at dark with a flashlight and look at all of Cushing's brains. It sounds like a very good, like kind of Halloween thing to do here. I think, his science, what Harvey Cushing did was pretty amazing. So he had this huge brain bank, which is now part of the library. It doesn't look like this. It's all cleaned up and nice now. And his science really was pretty spectacular. But I think us in 2022 here now really want to kind of look at it, not necessarily in a glass jar, but can we look at it like in living people, right? Or in living creatures. And there's different ways to do that, right? And I know that Martinos Center has expertise really in a lot of different areas, right? So MRI, EEG, TMS, and PET. But really, and they all have great assets, and they also have some limitations, right? So I think they're not necessarily exclusive at all. But today I'm going to focus on PET, right, or positron emission tomography, which some of you I know have been in the field longer than I have over at Martinos, but some of you folks maybe not. And so really, essentially, what PET does is it takes a look at I always tell my subjects and patients and things, it's like a little bit like a medicine, essentially. And then we radio label it, right? There's different ways to do that. Carbon 11, F18 are the most common ones, really. But it gives us an ability to take any kind of molecule, if you would, that can get into the brain and behave nicely, to be able to put some kind of trace on it, essentially, so we can get some kind of detection. And that's what this thing is over here. This is the molecule that's there. And it gives off an electron due to I think it's the weak magnetic force, and then it gets annihilated essentially when it finds an electron, and a gamma ray is given off, right? It's a small amount of energy, but that small amount of energy can be detected. And that's really kind of, to me, the core of what PET does, right? In PET, just one slide, I guess a shout out to the kinetic modelers. To me, it's fairly complicated here, but there's a lot of different processing steps that are involved here, right? So the mathematicians or compartment modelers look to see which kind of model fits best for each tracer, right? Each one of those molecules, as we call it, a tracer or a radioligand, really has to have all this done kind of for each one to make sure that these things are behaving how we think they are. So they do things like compartment modeling, they do time activity fits. Often we do parametric images, but really kind of the focus for us here is the end result, right? And the end result is in different parts of the brain, you can have things like VT, which stands for volume of distribution, or BPND, which stands for binding potential non-displaceable. But really just think about it as like, densities of a certain either target. It could be a neuroreceptor, it could be a molecule, it could be something else. And I think this part of it here gives us the ability to do quantitative imaging, which I think is very exciting, because if we can do quantitative imaging, then we can look at different kinds of conditions or disease states and understand, is that important in that condition, right? I guess the other thing you can do is really cool pictures. Maybe not quite as cool as DTI. I think that wins for the coolest looking stuff, but PET to me is maybe a nice second one there. And this is just in general, this is actually different tracer. This is 5HT1B that we did years ago, but it gives, I think, a good estimation of just what you can kind of see and expect in PET imaging. You can see that it doesn't do a great job at anatomical delineation. Because of that, we often, almost always really, use MRI to co-merge those, so we get a little bit of the best of both worlds. Okay, so you mentioned before, and by the way, I don't know what the Martinos kind of central rules are, but I love people blurting out questions or even answers. And so at any step of the way, please just go ahead and shoot out some things, and I'll be very happy to try to answer them. And so at the Yale PET Center here, I guess I feel really fortunate in the sense that we have a lot of these tracers to be able to look at, right? And so we talked about a little bit before, but you can look at a lot of different things, right? These have all been validated to different degrees. And so you can look at things like dopamine or glutamate, certainly serotonin, translate protein or neuroinflammation. We were just discussing a little bit before, cannabinoid receptors or CB1. But today I'm going to focus on this other one, which some of you might have heard of, called UCBJ. And that really looks at a thing called SV2A. So we're going to talk a little bit more about that. Oops, get on the right screen. There it is. And so this is the actual molecule. So UCBJ, you can see the carbon 11 molecule here. So this is the tracer part of it. The backbone of it essentially comes from the levetiracetam, also known as Keppra family, sometimes called piracetam family. And so the chemists here really did an amazing job, along with UCB Pharma over in Belgium, originally developing that. There's been some iterations and even some different kinds or versions of this, like an F18 version of it. But I think the core basic part of it is that, yeah, this molecule might give us an ability to look at synapses in living creatures. Oops. And so one question might be, who cares? Why do we need synaptic density, or who is interested in synapses? And there's some examples here like Alzheimer's and autism and depression. But as a neuropsychiatrist, I've done this game with myself trying to think of, can I think of a disorder in the brain that doesn't involve synapses? And I just can't. I think they all involve synapses, maybe even sometimes indirectly. But at some point, that's kind of like the final common pathway. And so I think that's where it gets very exciting to look at synapses in living creatures to say, okay, how do any of these diseases affect this, right? And so maybe the second question is, okay, whoops, my little cursor here seems a little anxious, but, and so we're gonna look at this with, so, what is the SV2A molecule? So it stands for synaptic vesicle glycoprotein 2. And really, if you look on the left side, this is just kind of a cartoon diagram of a synapse. And so you have the presynaptic side or presynaptic neuron, and the postsynaptic one. In the middle, of course, is the synapse, or the gap between neurons. And really, it doesn't really depend actually on what kind of neuron it is. They all use a similar process of packing all of their little goodies, right? All the little neurotransmitters into these vesicles. And these vesicles go and they bind to the presynaptic side, and then they basically open up and release whatever those neurotransmitters are. And so if we were going to take a blow up of this synaptic vesicle here, you could see this here in this cartoon. Synaptic vesicle, or SV2A, is this transmembrane protein that's right here. So it's involved mostly in exocytosis and endocytosis. I think if some of you folks are really well versed in the preclinical models, synaptophysin is a similar molecule. It's been really kind of the gold standard of kind of like synaptic vesicle or synaptic density research to this point. We've shown in previous work that synaptophysin and SV2 are pretty well correlated. I'm not going to show that data today. It's more of a clinical focus. But I think it's a nice kind of translational piece to look at synaptophysin and know that SV2A is probably going to behave pretty similarly in these different kinds of disease states. And so for these disease states, we can look at a lot of things. And so this first work came out was in 2016. Now it's been that long in Science Translational Medicine. That was the first one that was in healthy controls. But since then, we've really expanded and looked at a whole host of different kinds of conditions. And so not all these have been done by us. Huntington's disease was done over in Leuven in Belgium, but basically all of these have been looked at in some degree and even more so. But today I'm not going to focus on all of those kinds of things. I'm just going to spend time on one particular condition. And that condition was originally described by, interesting enough, a geologist who was also a physician. And his first time that the condition was described was in this essay on shaking palsy. So the shaking palsy was the original name of it. Of course, afterwards, it became Parkinson's disease after James Parkinson. That was in 1817 when he was living in London. And so what is Parkinson's disease overall? In a kind of a very basic textbook, medical school class, will say, hey, this involves dopamine and involves it in a thing called the substantia nigra, which is this black substance, literally, that's in the midbrain there. And it's a decrease in that substantia nigra that's there. And so often the clinical signs are things like bradykinesia or slowness of movement, resting tremor, often on one side. Those are kind of like the classic hallmarks of that. Well, I think now we know a little bit more than that. We know that, in fact, Parkinson's is more than just a movement disorder, and it involves a lot of other things. In fact, a lot of times the presenting symptoms are not movement-based, but other things such as sleep. There's a condition called REM, sleep disturbance, or RBD, and often the people that have that tend to have Parkinson's later. So it's almost like a pre-Parkinson's, and depression. And so, depression is common in a lot of different folks across the gamut. But if we go back and look at medical histories of people with Parkinson's, a fair amount of them had depression that happened beforehand. And so just as that's been true, we also know that saying that Parkinson's is a dopamine condition is true, but it's not the whole truth. Turns out there's a lot more, right? And so if we look at the pathogenesis of Parkinson's disease, it's really alpha-synuclein, right? So that's one of the proteins that makes up a Lewy body, classic pathological hallmark of Parkinson's disease. And we know that that it happens even before that stage. And so it looks like really in the synapses, there's synaptic degeneration that really happens very, very early. In fact, there's probably synaptic alpha-synuclein deposits that are really driving some of that behavior or some of those conditions. And so just as we know that that's been really kind of happening in the brain, we know that it doesn't just get regulated to dopamine areas. In fact, it happens in all different parts of the brain. It might just be the fact that the dopamine neurons, for whatever reason, are a little bit more particularly sensitive to these kinds of damage. And there's been a fair amount of genetic work, so alpha-synuclein itself, but also things like Parkin, DJ1, PINK1, LARC2. These are the most common variants that have been found to be involved in Parkinson's disease. Most of Parkinson's is still idiopathic. We don't know the cause of it, but of the genes that we have, these are the most common players. And if you look at all those genes and try to say what's the common link behind those things, you got to argue that synaptic loss or synaptic machinery really is that. And so that's kind of our journey here, right? So the first work I'm going to take you through is what has already been published. It was the first paper that was involved in Parkinson's disease with this tracer. And so let's look at the methods here. I don't know if there's any questions up to this point. No? All right, we'll keep trucking. And so, like we said before, we're really going to combine MRI and merge this with a PET scan. These folks had at least 60 minutes. Really, most of them had 90 minutes, but at least 60 minutes with on what is today still the highest resolution PET scanner in the world, the HRRT. Hopefully that will change soon. Both of our centers are working on next generation machines, and I can't wait for that to happen. This one, HRT, is over 20 years old, so it's high time to get something new here. We did do arterial blood sampling. I'm not really going to show that data, but it correlated really well with our reference model in general. So I'm really just going to present the BPND, or the binding potential results, for all the PET folks over there. And so we looked at a couple different areas, really, right? So we're going to focus on the motor circuit in here. So we're going to look at things like the striatum, substantia nigra, but also like motor cortex, pre-motor cortex supplemental, those kinds of areas, along with some exploratory ones, just to make it interesting. We did control for partial volume effects when we could. One of the interesting things is when you look at areas as small as the substantia nigra or other brain nuclei, there isn't a great way that I've ever heard of for trying to do a gray matter mass there. So those ones are uncorrected. Okay, one tissue compartment model for those other PET folks there. And so this initial sample had 12 and 12. So we had 12 Parkinson, subjects or folks with Parkinson's disease, 12 healthy controls there. And then I think, the years of diagnosis, relatively soon, 4 years, but some of the folks had as least as 2 months, some of them as long as 10 years. There's a clinical staging called Hoehn-Yahr that goes up to 4 stages. And essentially all these folks are stage 2, which means that they really just have mild bilateral impairment there. So that's basically all the same. And then there's the field years ago decided they're going to do a standardized rating scale. Technically, this is called the MDS-UPDRS, but the Unified Parkinson's Disease Rating Scale. This just gives us a big kind of estimate of all the kind of clinical characteristics you would have. So again, this is mild bilateral. Maybe UPDRS is a little bit more moderate than that, but these are certainly not the most severe folks, and not necessarily folks who've just had it, and are asymptomatic or mildly symptomatic. It's somewhere in the middle there. Just in general, in Parkinson's disease research, we try to hold any at least dopamine medications during the scan day to reduce potential compounds. Not clear that would happen in SV2A. It's not clear it wouldn't either. In DAC, clearly that would affect those kinds of things. And so we just did that as kind of a standard of research. This was the first scan. Somehow my labels fell off here, but if you look at the bottom one, that was the first Parkinson's disease patient scan with this UCBG tracer to my knowledge. The middle one is a healthy control, and then you have an MRI to show kind of a comparison there. But let's look at the quantitative data. That becomes the most interesting part. And so in this sample, what we did was we kind of pulled some folks, or pulled the different regions apart and looked at brainstem nuclei, looked at the cerebellum, subcortical regions, and then the cortical regions. And I think the thing that I found kind of striking is that there are decreases all over. They're not all statistically significant, right? And so in this sample size, most of the brainstem nuclei are significantly different, right? So you can see the locus coeruleus. This is kind of the origin of norepinephrine. The red nucleus is involved in a lot of movement. It's also pretty high in serotonin and GABA. So those are both decreased. The parahippocampal gyre is kind of an interesting thing to find there significantly, but there's some work that there's alpha-synuclein buildup even at an earlier stage around that area. Kind of interesting enough, maybe putamen, which one would think of as having large decreases in Parkinson's disease, didn't show. It was decreased, but not hugely significant. There's possible reasons for that, one of which is that, even though we think often in the dopamine world of putamen being really highly innervated with dopamine, it's really just a minority player there, as opposed to substantia nigra. Now, our substantia nigra template, I have a homemade template, really, and so it doesn't distinguish, and we don't have the spatial resolution. Maybe we will with these new scanners coming out, but we'll be able to tease out the pars compacta from the pars reticulata. It's just kind of one region in there at this point. And so even with that said, it's still highly dopaminergic. I mean, I don't know exactly, could argue 30 to 50% around there would be dopamine. And because we don't find Parkinson's without substantia nigra involvement, I think it was encouraging from the research point of view to see such large decreases, around 50% in the substantia nigra. We just kind of focus on that and blow that up. And this, I think, gives you a good estimate if you can look at these small little, or these white arrows here. These are the 3 different actual ranges here, but you can kind of see that kidney bean here. And that's the substantia nigra that kind of drops out there. And again, you can see it on this side and there. And so I think that gives us a good estimation of the really pushing the boundaries of what the spatial resolution that we can do today. But it also gives a nice visual estimation of what happens in Parkinson's disease. I was excited about this finding a lot. This is NC, normal controls, or you can think of as healthies. These were age matched. So those lines there, that's what they designate. So these are folks that have the same gender, the same age, and overall the same BMI. And overall, except for that one, my second to last one, they really correlated really well. And so you can see those decreases overall. And so that got me excited as a clinician, thinking, well, if you want something in a clinic, you want something that has that kind of specificity or sensitivity, rather. We could really kind of tease that out. At least in this sample size, it looked quite well. We're lucky enough, this work was done with our really friends and collaborators over at AbbVie, and they had access to some postmortem data. And so this is not the same subject sample, completely different subject sample from a brain bank. Boehner actually over in Arizona. And so what we could see though overall is that in these, presumably much more advanced Parkinson's patients, you still see the substantia nigra being decreased over here. And putamen, kind of similar findings, decreased but not necessarily significant. So that's something that might be interesting in SV2A. Cortical areas were also there. And this, my next slide, I'll describe in a little bit more detail, but maybe a little bit more interesting than I thought. There's debates about, how does Parkinson's disease progress? And so at least in this sample size, we did see some decreases. They weren't always significant, but just overall, we found some decreases in the SV2A and cortical areas. And so alluded to this in the last slide, and that is just, how do we pathologically think about the progression of Parkinson's disease? And Braak staging of the same Braak as Alzheimer's also did a Parkinson's disease staging. That's really based on Lewy body and alpha-synuclein staining. And so the argument there, or the discussion piece there, is that, hey, listen, in the very early stages, this happens probably in the brainstem nuclei. It might even happen earlier than that in cranial nerves. And then it spreads in this kind of, general brainstem to cortical distribution, right? So it starts to come up through, brainstem into midbrain area, some cortical regions, parahippocampal gyrus too. And then kind of in the later stages, it really is affected all over in cortical regions. I think that's one of the things we'll keep kind of coming back to in some ways in the talk, is just thinking about, can we find an in vivo version of that's true? And that's an open question. Lucky enough to have a lot of friends and colleagues here. And so our colleagues over in Europe, one of these is in Leuven. Actually, Koen van Laar is with us right now in sabbatical. He's just really wonderful scientist in person. And so he was one of the groups that led some of these other papers that really we all published about the same time, along with folks in Aarhus and over there, our friends at in vitro in London. And so there are some differences between these findings, but overall we found some similarities too. And clearly the substantia nigra being decreased is one of them. The percent difference was a little bit different there, but overall that was a common finding of that. Some follow-up work I can say is that, we're kind of in the beginning stages of seeing, can we pull some of our data together to really have a better kind of, appreciation and sample size to investigate this a little bit deeper to see what our similarities and differences are. But on this talk, I'm going to focus more on just kind of updates that we've had here at the Yale PET Center since that first paper. So that's really the next stage of the talk. And so we've done pretty well here. So we went from a sample size of 12 to 28. Truth be told here, we probably have some more than that right now, but these are the folks that have been analyzed at least as of a month or two ago. And that's with the SV2A. In addition, what we've done is really added DAT scans. The Leuven group has done this too. And so DAT stands for dopamine transporter. And we talked about the importance of dopamine in Parkinson's disease. And In fact, it's FDA approved to have a thing called a DAT scan, which is a SPECT version of a dopamine transporter. And so this newer one that's been used in Europe for some time now, actually even used clinically, is a FEPE2I, which is kind of would I'd probably argue a little bit more, sensitive, improved version of a DAT scan. So similar to a DAT scan, it does a good job looking at subcortical areas, but not so much in the cortical regions. Actually, not at all really cortically. And so of those, it's a subset. We didn't get all the Parkinson's folks to do that. We got the majority of them, and you can see overall the total UPDRS score. These folks have had Parkinson's disease a little bit longer, and that's kind of what the next data set's going to really have, right? And so in some ways we can pull this out. I think on one of the other sides I have the exact sample size number. But basically what we did was said, okay, let's look at our same healthy controls and let's look at Parkinson's disease. But what happens if we pull this out a little bit and look at folks that are really more recent diagnosis, under 2 years is often what's used in the literature. And then more, people with more moderate disease is probably fair to say, and that's over 6 years. The clinicians might kind of discuss that with me and say, is that really the most accurate version? I think with the data that we had, that it made sense for us to kind of make that mark there. But if we look at this and look at substantia nigra and we focus on that region again, you see, I think what you would kind of expect to see, which is encouraging in some ways, and that is you see really healthy controls, essentially 1 BPND, right? So this is just using a reference region that's kind of like the standard, and you start to see this kind of sequential drop. And so early PD, you see a decrease around maybe 20%, and then a little bit later stage PD, you start to see about a 50% decrease from that mark. And so that, I think, is exactly scientifically what you expect to see. Brainstem follows suit. Pretty much every region follows suit in some degree, right? Even in the cortical area. So it's kind of interesting. Again, not statistically significant in most areas. If you lump these with all the PD together, that's on the right side, then you'll start to see substantia nigra, brainstem, even the caudate, interesting enough, and putamen are significant here, but not in the cortical areas so much. So again, subcortical more affected than cortical overall. If we focus on the cortical side, and so these are kind of a little bit more exploratory regions, right? But if we just kind of take a look at the cortical regions overall, same kind of pattern, but not necessarily significant, right? So that might hold true in kind of in a Brock staging, where again, you start to see subcortical differences sooner than the cortical activity. And this slide really kind of just pulls that out even more. So if we just compare it to our healthy control population, you can look at the early PD. Yes, you see the subcortical areas really becoming significant there, and some decreases that are there. And then when you see in the later stages, you see quite large differences in the subcortical region. So that expands, but then you start to see the cortical regions overall. So this is similar to Brock staging. I'd argue the one difference or subtlety might be is that I don't know if Brock staging predicts any cortical activity involved in this. I think one of the difficult things is Brock staging is a pathological kind of term, and we're looking at it from a clinical perspective. So that might not be exactly comparable in that sense. We look at just in general, what happened there? There it is. If we look at just correlations here, right? So we're not going to pay attention to necessarily years of diagnosis in the graphs onto the right. But if we look to the left, just at the actual clinical data, what we see is UPDRS. So that is just part 3 is a motor score. So that's often used in a lot of Parkinson's clinical trials. And overall, that's significant, 0.41. So that to me just says it's not perfectly distributed clearly there, but overall our sample size seems pretty congruent as we expect to find. And then if we look at that same part 3 motor scores in areas like the substantia nigra, you do see a correlation there along with the red nucleus. And so again, that kind of behaves, I think, overall how you would hope it would be. The more severe the symptoms, the more the decrease in synaptic density. That makes sense overall. If we compare early versus later PD, you can see just between those two, there are decreases there. They're not significant. Part of that might just be these huge standard deviations kind of coming through here. The fact that our sample size is pretty small so far. So as time goes on, we're maybe able to study more of these differences. I wouldn't be surprised to see some of these things kind of coming online there. Okay, if there's no questions on the SV2A world, I'm going to switch gears a little bit into DAT. Okay, so for DAT scans, and so this tracer has been fairly well published, certainly more than UCBJ. And I think overall, most of the folks find similar things to what we do. And so these two graphs, left and right, really the only difference there is the reference region. There's the cerebellum that's used. The cerebellum, I've had this discussion, the cerebellum is involved in Parkinson's disease. To the point and the extent is always open to debate. But and so we looked at it with cerebellum as a reference region, which is the most common way to look at FEP2I. We also looked at the occipital region. It doesn't matter overall. It seems like you have pretty similar findings. So at least in this case, it doesn't, I would argue it's probably not terribly important. But overall, you do see large decreases. And again, these are kind of mixed between late and early or moderate Parkinson's disease statement patients, but you can see overall, yeah, large decreases basically everywhere. So not terribly surprising that a more dopamine specific tracer can do a really well job, probably a better job than UCBJ, at least at the earlier stages. I think there's still questions I have with this, and that is, as time progresses, is there a floor effect here? And maybe there's not much more dopamine to lose, and then it might not do quite as well, but we'll hopefully get an answer to that question at some point. If we look at the clinical motor scores, so same as before, you can also see really nice correlations. Putamen does really the best job, just looking at the R.71. Substantial nigra and caudate also kind of come up there, at least significant or close to significance in there. So overall, DAP looks like it's tried and true. It's well established. This tracer looks like it's really good at least being able to tease out, maybe the early to moderate stages of Parkinson's disease. Compare them. Look at total UPDRS, you can see kind of the differences there. So the UCBJ does a pretty good job there. Probably FEPDRS is going to do a better job, just overall looking at duration and total UPDRS score. Both these things I think are always tough in any clinical field. When you look at things like duration, usually when people say that, they're looking at duration of diagnosis or duration of symptoms. But both of those things can be a little bit tricky, right? Because some folks I have seen over the years where, they might have symptoms for a week or two or a month, and they or their partner or family are really observant and they get in and get that diagnosis really fast. Or some folks might last 10 years. And so there is a lot of fluctuation in there. I think that's part of the reason why, just not in neuroimaging in general, but just in the field, there tends to be a lot of noise in those kinds of variables. And so, I take all these things with a grain of salt. That being said, they look pretty good overall in this case. We look at the comparisons, right, of FEP2I or DAT scan versus SV2A. There's going to be it's kind of like a heat map that was done by Praveen Hanar, one of the grad students of Rich Carson, who a lot of you know. And so he did a great job with this. But overall, when you look at this, you can see like the heat map here, you can see in the substantia nigra, that is probably the biggest overlap, which makes a lot of sense because there's a lot of dopamine there. So that kind of holds true. Other areas, though, you still see some significance pulling out, and things like the caudate or in other areas too. Okay, I guess an important note here that this was in 10 subjects that were within a short duration. We're still working overall on trying to get a larger sample size on this. So this is getting to be a work that's still going on at this point. Speaking of that, there's a lot of work going on with this. And so really, I think the next steps for this overall are comparison between Parkinson's disease stages. And so early, mid, and later. Lucky enough to get an R01 relatively recently. And so that's going to be a big part of that work. It's just trying to understand, okay, are all these tracers kind of equally good at different stages or equally bad? Are there different kinds of things that pop out at different stages? How does that work? Another thing I think is really important to understand, I think this is important when you start thinking about things like drug discovery, is, okay, can we detect longitudinal differences, right? And so if we scan these people, which we've already done, I can't present the data now, it's not really polished enough to do that, but we have a fair amount of folks who have come in after a year to 2 years. And so we're going through those processes right now, just trying to understand, hey, listen, are there differences that we can detect with either one of these tracers? I think that'd be really, really attractive and important when we're starting to do drug studies, because there is so much variation in things like clinical symptoms or disease duration. It'd be nice to have an objective measure. So that's something hopefully something like PET could help out with. And so, at the end of the day, sometimes when I think about this stuff, it's like it's kind of like a horse race, right? So we have DAT scan on one side, we have SV2A on another. We have a lot of the MRI modalities. I haven't really presented that or discussed it at all, but we're doing things like neuromelanin, resting state, NODI, looking at things like neurite optic density imaging. And so we're putting all these together to see really, is one of these better in any kind of way than any other one? Or maybe it's a cumulative thing. Maybe if we are clever and can figure out ways to merge these things in a certain way, maybe there's a composite score, something similar that would be even more beneficial. So that's kind of work that needs to be done, and it is going on now, along with overall just ROI templates. It's kind of in some ways maybe more mundane, but it's actually really important. And when we look at these different brain areas, say the substantia nigra, there's no definitive way to really define those kinds of things. And so we're looking at different kinds of templates and different ways to do these things to see, okay, can we come up with a possibly a better way, or maybe a little bit different way in the field to kind of measure those things. I think overall, I am a clinician still. And so at the end, to me, I'm like, when I think about this, can this stuff be useful? Can something out of all this work be important for patients or something in the clinic? That really is my goal, not just in this project, but I'd say really just overall in my career and what I focus on now. Okay, what are we? We're 12:40. I think I talk fast here. But always the most important slide here. So I'd like to thank, really the whole PET Center. So there's so many people here. Abby Vanima, who was with us years ago, but went over to Abby. He was really the first funder of this. Wouldn't have been at all possible to do it without him. He's also a great scientist, and so it's really fun to work with him in that capacity. Shirley Tanaz is the movement disorder neurologist. I'm not one of those, and so she really does just an amazing job at looking and at all these folks clinically with a detailed eye, making sure this is exactly accurate. Also, a discerning eye for the data, so that's always appreciated. Praveen Hanar, we spoke of before. Sophia Holmes is a psychologist who's interested in Parkinson's disease. She has this R21. She's really looking at depression and Parkinson's disease, trying to tease those things out. So that's work to be done. Ying, who is my own postdoc, and which I think a lot of you know. Just as important, if not more important, Cure Now. One of the things I love about working in this field is really working with the community in Parkinson's disease. They're just really, really spectacular and really well organized. And so the organization we use in Connecticut is called Cure Now. And we go to social functions with them, golf tournaments. I really feel like, I hope they do too, that really it's just been a team approach. It's both of us kind of, or all of us working together, trying to see, can we do better in any capacity? That's all I have. Thank you very much. And please give me some questions.